What's up, everybody? So in this video, we're going to be talking about oogenesis. This is the topic you guys most likely just hate, but trust me, I'm going to make it as easy and as clear as possible. So remember in the last video, I asked you what is the difference between Eminem and Miley Cyrus? And the answer is Eminem has balls and Miley Cyrus does not. So what is so special about balls or testes? The testes can make sperm, and this process is called spermatogenesis, and I made a whole video on this, so you can go check that out if you want to. Now, what does a female have then? A female's version, the female version of balls is basically ovaries, and they are responsible for making eggs instead of sperm. So we call this process oogenesis, and this is what we'll be focusing on in this video. Now, once this egg and sperm is made, they can meet, so the sperm can meet the egg in a process called fertilization, and I'll make a whole separate video on this. Now, once fertilization happens, so now this egg has both the female, the mom's Miley Cyrus's DNA, and now the sperm donated the male's DNA. So now this egg has both DNAs to be able to develop into a normal, healthy human being, right? This will happen during pregnancy. So let's just get into oogenesis. So what we're going to do is we're going to zoom into this little female reproductive system. So here we have it. So real quick, just because I can, I'm going to quickly label these things for you to have you caught up on the female reproductive system. But to do that, we got to slice this female reproductive system open so we can see and label all the parts. So here we go. We slice it open, okay? Because right now it's closed. It's three-dimensional. This is how you will see it if you were to look at it inside the human body. But we want to slice it open so we can see what's going on for real. So here we go. We're going to quickly label some stuff real quick. So first, where do we start? Ovary, okay? We're going to start at the ovary. Why? Because the ovary is the female version of the testes. This is where what is made? Our lovely little egg, right? This is oogenesis. Oogenesis is going to create this egg for us. Now the egg is released from the ovary into what? This little pipe. What do we call this pipe? The fallopian tube, otherwise also known as the oviduct. Now, the egg is going to wait right here for what? It's going to wait for sperm. Sperm is going to be ejaculated into what? The vagina, right? We call this area here the vagina. Now, after it enters the vagina, oh no, that's wrong. I labeled the wrong thing. Give me 1 second. We call this right here the vagina. And then it's going to pass through this little narrow opening here, which we call the cervix. After it passes through the cervix, it's going to go into this area here, this hollow space here called the uterus. Now, it's going to swim all the way to the egg, because men's got to chase the woman, right? So the sperm is going to go chase the egg. It's going to fertilize the egg here in the fallopian tube. Now that the egg is fertilized, it will travel all the way down here into the uterus. Specifically, it's going to go anchor like a seed into this wall. This wall is called the endometrium. And now here it's going to grow and grow and grow in this wall all the way till it's a baby, right? Till it's a full-grown baby. So this is like the basic, all the things you need to know for the structure of the female reproductive system. But did I explain oogenesis? No, right? So let's go into here, the ovary. You're going to zoom into right here and see exactly how oogenesis happens. So here we have it, the ovary, the fallopian tube right next to it. Now, let's open up this ovaries to see what's going on the inside. So we slice it open to see what's going on the inside. So right now there's nothing, because I'm going to build you a diagram and hopefully it will make a lot of sense. Now, what is this right here? This right here is a little vessel, These vessels are very important. Why? Because we have vessels everywhere on our body. If you cut your knee, you're gonna bleed, right? If you cut your face, you're gonna bleed. If you cut anywhere, you're gonna bleed, because everywhere there's vessels. Vessels are super important because they carry a lot of important things to the area to supply it. It's like a little messenger. It's gonna bring nutrients and everything that all the areas need. So just bear in mind, there's always vessels everywhere. So where do we start? So I'm going to explain oogenesis first in terms of just all the key steps, and then we're going to look at the nucleus, and we're going to look at what happens to the DNA. Okay, so we're first going to look at the broad view and then the specific view. So if you don't get it at first, do not worry, because we're going to get to it. We're going to get to it nice and slow. Try and make it clear, because if I rush this, you're going to be all confused, and you'll just have to watch another damn video. So let's just do this slowly and properly. So it all starts with this cell here. Okay, we're in the ovary. we have this little teeny tiny cell, right? What's this? The nucleus, here's the cytoplasm. It's an ordinary cell. This cell we call an oogonium. It's a little stem cell, right? What is a stem cell? A stem cell is any kind of cell that has the potential to become any kind of cell, right? It's like a fresh young baby. It can become an actor. It can become homeless. It can become literally anything, right? It has the potential to become anything. So that's the same thing here with the stem cell. It has the potential to become a full-grown, mature egg cell, right? But it's going to take some steps to get there. So we call this an oogonium. Now, this oogonium is going to get transformed into another kind of cell. What's this cell? This cell right here is what? Called a primordial follicle, okay? Primordial follicle. What's different about this one? What's different here is that it's not just one cell anymore. Now we have this like soon-to-be egg cell, this oocyte, but it's surrounded by all these teeny tiny cells. So the combination of this teeny tiny oocyte and all these other cells is called a follicle, a primordial follicle, right? So it's not just one cell anymore, it's now a follicle. But we only care about this little cell inside here because this is the thing that's going to be released later. So, but bear in mind, the combination of these is the primordial follicle. Now, the primordial follicle is now going to be converted into another cell, okay? Notice here, it's very similar. Very, very similar. We still have the oocyte and we have all these granulosa cells, but you don't need to care about the name of these granulosa cells. Just know that there's this thin layer of cells surrounding this oocyte. Now we call it what? We call it a primary follicle. The only difference really is that these surrounding cells grew a little bit more and they're a little bit more tightly packed, but you don't need to care that much about that. So now what's next? Now this primary follicle is going to develop into something else. Specifically, it's called an early secondary follicle. What's different here? Two things. It's got way more of these surrounding cells, way more, right? So it's growing, it's getting bigger and bigger. And we've also got this light blue layer. What do we call this layer? This is called the zona pellucida. Zona pellucida. Basically, this layer is very important later in fertilization. So you'll see this name again. That's why I brought it up. Because the sperm has to penetrate this layer to get to the egg. So that's very important, this layer, the zona pellucida. Remember that. Okay. Now, this early secondary follicle is going to develop even more. It's going to become what? What do you think? Early? Now late, right? It's going to become a late secondary follicle. Late secondary follicle. Now, this late secondary follicle, what's different? It's pretty much the same, except now there's all these empty spaces that has like liquid in them. You see? We call these empty little spaces antrum. Okay, so this is one antrum, this is another antrum, and you'll see why these antrum are forming. But that's basically the only difference between the early and this late secondary follicle in terms of the looks, right? It still has the zona pellucida, but that's it. That's the only difference, really. So now, this late secondary follicle is going to become another thing. What do we call this one? The Graafian follicle. Okay, the Graafian follicle. So what's going on here? This part might confuse you a little bit, but it will make more sense later in this video. So let's first look at the obvious part. Notice all these little antrums, right? Look here, now we have one huge antrum. All the antrum here fused together. We had all of these, we had 1, 2, 3, 4, 5. They all fused together to form one huge antrum, right? So that's one key difference between the late secondary follicle and the Graafian follicle. Now what's another difference? Remember that oogenesis involves meiosis, right? Meiosis 1, meiosis 2. We're going to get to that a bit later in this video, but just bear that in mind. So what happened between this one and this one? What happened between the late secondary follicle and this Graafian follicle is meiosis 1. So the cell divided. It actually divided, but it's very hard to see. Because here we had one cell, here we have two, okay? Now you might be asking, okay, where's the second cell? We have this one cell here, and then this other teeny tiny cell. So it divided. This is meiosis 1, so we formed two cells here. So this is what happened from here to here. Now, this little teeny tiny cell is called a polar body, and it's gonna leave. So basically this cell here, split into two, the polar body and then this cell, right? So now this cell will look normal again. So this late secondary follicle became this Graafian follicle by dividing into two cells. The small cell called the polar body left. So this is just a normal cell, it's just super tiny. This one overwhelmed it. It's like putting two people into a room full of food, right? The dominant one will be able to survive and eat all the food and keeping the small, weaker person away, and the small, weaker person will never develop, right? So that's the same thing here. So one of the cells were much stronger and developed, and the other one just kind of got sent away. Okay, so that's difference. Now after this little space here is full of fluid. Pay attention to this. This little space here is full of fluid, Now this fluid keeps building up. So you can imagine there's a lot of pressure building up. It's when you keep filling up a bag, eventually it's gonna burst, right? So it's the same here. You keep filling it up with water. All of these little cells here are synthesizing water, and it's filling up, and eventually it's gonna get so pressurized, it's gonna pop out this cell. It's gonna shoot it out. Into where? The fallopian tube. So this is then our egg cell that is going to go and get fertilized with the sperm, right? Wait one sec. Okay, so what do we call this? So essentially, we formed, when this was in here, a polar body. Let me bring the word here for you, a polar body. And then the remainder, so after this whole egg gets ovulated, we call that ovulation, right? When the egg is released, ovulation. So when the egg is released by ovulation, now we have this polar body left, and we have this thing here. This thing here is going to become what we call the corpus luteum, and it looks like this. It's going to start changing and degenerating and forming this corpus luteum. But this corpus luteum has an important job later during fertilization. So you'll definitely see this name again, the corpus luteum. It's called the yellow body. Corpus means body, and luteum means yellow. So it's the yellow body. Okay. So that's basically the process, right? So obviously we're missing some stuff. We're missing what happened here at the nucleus. We're missing what happened here with meiosis. But overall, the basic steps is we start off with the stem cell called oogonium. It develops into a primordial follicle, which then develops into a primary follicle, which then develops into an early secondary follicle, and then a late secondary follicle, and then this Graafian follicle, which will release the mature egg, and also this little polar body, and then the rest of it will go into this little thing here called the corpus luteum, which has an important function also, but we'll talk about that later. Okay, so let's go into slightly more detail. So here we have the same diagram. I'm just going to fill in slightly different things, okay? More about timing and about the DNA, the nucleus, okay? Because that's very important. Because remember, just like spermatogenesis, which involves meiosis 1 and meiosis 2, right? Because meiosis is the process of forming gametes, right? Like egg cells and sperm cells. So right here, meiosis is happening. I just want to explain it a bit more clearly so it's more obvious to you. So basically, remember in a male, this is very important to understand. Remember in a male, spermatogenesis only started after puberty. Only started after you're making testosterone and these kind of things. Only after puberty, right? Very important. In a female, oogenesis happens when they're still in their own mom, when they're still a fetus. Isn't that crazy? So a female starts oogenesis way before puberty, right? Whereas a male starts at after puberty. So this whole process of converting this oogonium, right, this first cell, the stem cell, into this primordial follicle, this happens before puberty, when the girl is still a fetus inside her own mom, right? So when she's just starting to develop herself, okay? Now, this will be called, this right here, this cell here, this primordial follicle will start meiosis, okay? It's going to start meiosis 1. So we call it a primary oocyte because it's starting meiosis 1. So all of these primordial follicles will be built up pre-puberty because you're going to make thousands of these. So by the time the girl reaches puberty, they will have like 300,000 of these primordial follicles, right? And then these primordial follicles will then start meiosis, okay, meiosis 1. So for example, this one here will start meiosis, but it won't complete it. It will stop in prophase 1. So I'm going to put this here inside the nucleus to indicate that it's stopping in prophase 1, P1. Okay, so we call this oocyte in here. Remember, this is the whole follicle, but the cell inside we call an oocyte, right? This whole thing here is a follicle, but the cell inside we call an oocyte. So this primordial follicle, this oocyte here, will start meiosis but stop in prophase 1. Prophase 1. So this is, at this point, we call it a primary oocyte because it is in meiosis 1. So the 1 for meiosis 1, okay? So it's a primary oocyte. Then puberty will happen, and we will convert these primordial ones into primary follicles, right? But meiosis doesn't continue. Meiosis will in fact still remain in P1, so it's still going to remain in P1. So all that's happening is the outside is changing, but not the DNA. The DNA is still in prophase 1. So we still call it, therefore, a primary oocyte. So we're still after puberty starts, this whole process will still keep going. So we're going to keep going, and the next one will be formed. But meiosis is still in P1, so we're still in P1, so we still call it a primary oocyte. Now we still continue, and we create this one, but it's still in P1, so it's pretty much stuck in prophase 1 or meiosis 1 until the last stage, okay? So we still call it a primary oocyte. Now, when this cell reaches this stage, right, the late, what do we call it again? The late secondary follicle, when it reaches this phase, now things are going to start changing in terms of the nucleus. So now we're going to finish meiosis 1. We're going to get converted from this cell into this one. Okay, so it's going to split, right? Remember I told you it splits. So we form this cell here, which will release. Remember, we split. So this cell here will now finish meiosis 1 and split into two cells. One cell is the polar body, right? And one cell is this one, the Graafian follicle, right? So now this cell is entering meiosis 2 because we just finished meiosis 1 to form this polar body and this cell. Now this cell here is going to initiate meiosis 2. So we call it a secondary oocyte. So this is where we have the secondary oocyte, 2 for meiosis 2. Now it enters meiosis 2, but again, it doesn't finish it. In fact, it's going to stop at meiosis 2, M2, meiosis 2. The only time when this cell, so remember this cell will now be released. Oh yeah, let me go back. So remember here, this cell is an M2. Let me come back here to make it a bit more clear. So this cell here is in meiosis 2, right? So we want it to finish meiosis 2, right? It's got to finish meiosis. But it will only finish meiosis if it meets the sperm, if it gets fertilized. Otherwise, it will end here and during the period, it will leave the body, right? With the period blood, right? So that's important to understand. So I hope this makes now a bit more sense in terms of the meiosis part. We start here, the cells will start meiosis, they'll stay stuck in prophase 1, which we call a primary oocyte, all the way until this part where it divides and forms a polar body and this secondary oocyte, which will initiate meiosis 2, but it will get stuck in meiosis in metaphase 2, and then it will get released into the fallopian tube and eventually meet the sperm. And once it meets the sperm, then it will finish meiosis 2. Okay, but this will make that part is not part of oogenesis. That is part of fertilization, which remember I said I'll make a video on fertilization, so that'll make more sense in that video. Now we're almost done here. Here we got a brain. Okay, brain is very important in many parts, right? So this whole process doesn't happen by magic. Remember, there's a part in your brain that looks like this. It's the pituitary gland. I'm sure you've heard of it. It's a little part in your brain that looks like a ball sack, but it's not a ball sack. And what it does is it releases little hormones, okay? These little hormones. It releases them into the bloodstream, okay? And the bloodstream will get transported all across your body to here, to the little ovary, right? Then they will leave these little capillaries, right, and have a job. The job of these two molecules is to stimulate this whole process to happen. Without these hormones, this process will not happen. So what do we call these hormones? FSH, I'll put it here, FSH, the orange one, and LH, okay? FSH stands for follicle stimulating hormone. That makes sense because it's stimulating these follicles to develop. LH is also important in this process, but it's more important in the letting go process. So I like to call it letting go, L for letting go. The process where this egg is where ovulation occurs and this final egg is released. So just bear in mind, both of these are very important, okay? And the release from here, a part in your brain. Now, one last thing that I think can help you a little bit, and don't worry, all this process I said in words, but I have a nice little diagram, word form diagram at the end where I write out the steps clearly in words so you can follow that and it should make sense even more, okay? Let's go here. So to make it make a little bit more sense, let's look at this. Okay, so before puberty, right, we had this little, let me make this more clear. I'm trying to make the DNA idea a bit more sense. So remember the primordial follicle, right? Which is formed before puberty, right? We formed this primordial follicle before puberty. Let's go here. Primordial follicle, right? It's going to start meiosis 1. Remember, I said it's going to start going into meiosis 1, and it's going to stop in prophase 1. Okay, so here we go. So this primordial follicle, it has all the normal DNA, right? So if we have a normal cell here, we've got whatever chromosome we look at, we always have one from the mom and one from the dad, right? It's the combination of these two that will give you your traits. So we call this diploid. When there's at least one from your mom and one from your dad for a specific trait, we call that diploid, or 2N, two variations, one variation from the mom and one variation from the dad. Now, before meiosis, we know the DNA duplicates, right? So it's going to form now two versions, two copies of the mom, two copies of the dad. But we still call it diploid because there's still only two variation, one mom variation and one dad variation, right? So this primordial follicle will undergo meiosis 1, but it doesn't complete it, right? It's gonna initiate it, it's gonna start, but it's gonna stop in prophase 1. Okay, so let's put it here. So it kind of starts it, but it stops in prophase 1. Okay, so because it stops in prophase 1, it's still in meiosis 1. So we call it what? We call it a primary oocyte. A primary oocyte, right? And at this point, remember how I said before you reach puberty, you build up like 300,000 of these. And throughout your lifetime until menopause, these 300,000 will get used up. A lot of them will just die from natural causes. So you may end up having like 1,000 useful ones, and the other 299,000 will just die naturally. Okay, they won't ever be released or ever be used. Now I have a picture for that later. Now, so it enters meiosis 1, but it stops in prophase 1. So we call it a primary oocyte. Now, puberty happens. So from puberty on, we know it's going to go through what? We know it's going to go through, from puberty on, this primordial follicle will undergo all of these developments, but it's going to stay in prophase 1. So it's going to become this cell here, this one here, this one here, but always remain in prophase 1 until it gets to a point after puberty, like from puberty on, where it finally divides into a secondary oocyte, right? So let's come here. So it's from puberty on, it's going to divide, it's going to develop and finally divide, right? So in between this cell here and this one here, there's a bunch of stages, right? A bunch of stages that is in prophase 1. So when it divides, it forms this what? You know the name, Graphian, right? The Graphian one. I can go back here to show you. This one, the Graphian one. Right? And this one we call a secondary oocyte because it's going to go into meiosis 2. Into meiosis 2. Now the other one, what do we call this one? Because it divided into two, right? They formed the Graphian one and this other one. We call this the polar body. Now, notice something important here. Now because this cell divided, so one cell got the mom stuff and the other cell got the dad stuff. So now we call it haploid or N because now there's only one variation. It's the dad variation and the mom variation. So we call it a haploid. Right, and this is the tiny polar body that I was mentioning before. Remember the little polar body? Yes, that's it. So now we're going to go into meiosis 2, right? So because this one is going to go into meiosis 2, it's going to stop at one of the stages. Do you remember? It's going to stop at meiosis 2, metaphase 2. So here we go. Maybe I'll put it, maybe I'll just open it. Okay. So it's going to go into meiosis 2. I'll put it here. Specifically, it's going to stop in metaphase 2. But because it's in meiosis 2, we call it a secondary oocyte. Okay, it's in meiosis 2. Okay. So we don't care that much about this. We care about the key one. So now, let's say eventually this cell is released. Remember, it stops in meiosis 2. We can go here. It stops in meiosis 2. It's released. And eventually, it's only going to finish meiosis when it meets the sperm. So let's go here. So now let's say fertilization happens. So finally, this egg will finish its division. Now, it only finishes this division, like I said, when fertilization happens. So only when the sperm comes into contact with it. Now, this cell will divide. And guess what? It will divide into one cell that has half the DNA and the other one that has half the DNA. But still, each of these only have one variation. So we call them N, or haploid, still. But this one, it's confusing, right? Because the sperm is going to bind to it and donate DNA. It's going to donate its DNA. So in reality, this cell will divide into two, but because the sperm released its own DNA in here, this cell here will have now the male and the female's DNA. So this cell will be in fact called a zygote, and this cell will divide and form a full-grown baby, right? So what can we get from this? We can get from one single cell, from one oogonium, right? From one oogonium, we can only form one mature egg cell, and all the other ones are polar bodies, right? So in reality, that's different from spermatogenesis. Remember, spermatogenesis, each one stem cell formed four sperm cells, but for oogenesis, only one is formed. The rest are polar bodies and useless, right? So, oh yeah, this is like that's basically the whole process, right? So you need to understand it in terms of two things, both the names of each of the stages, and then you need to understand what is happening in terms of meiosis, right? In terms of the stages. And it's quite simple if you just revise it a little bit. Now, like I said, the corpus luteum is going to have a very important role later in fertilization, not important right now. Now here is a little diagram that I find pretty interesting. So, if you're a girl, like I said, before you're even born, when you're a fetus, you make a lot of these eggs, right? These primordial follicles, you make a lot of them. So you can see you have over 1 million, you have like 5 million before you're even born. Now, once you're born, until puberty, you slowly lose these. So you have a certain amount before you're born. And then from there on, you just lose them slowly. And by the time you reach puberty, you have like 300,000, like I said. And then you'll keep losing them, partly because some of them just die naturally, and because some of them are released every month, right? Every month you have to release an egg. Now, eventually, you can see they decrease. Now, by the time you reach like 50, you have almost no eggs left. So this is the reason why females aren't fertile as long as men, right? Men can be fertile for a much longer period of time, a much longer age than females. Typically by 50, females have no more eggs left. And so they cannot release any more eggs, and so they cannot have any more babies. So that's why it's important for females to have children more early than later, right? Very important. Otherwise, they'll have no more eggs left. So keep in mind that a female's eggs decrease from before they're born only till they don't make any more eggs. They have a certain amount, and those will just decrease. Okay, so that's very important. You don't need to know this for IB though. It's just an interesting idea for you. Okay, so like I promised, here is the steps, the words that you can phrase this in when you answer the IB question. Everything is here that you need to know. And here is an exam question as well. So here's 8 marks. If you look at this, you'll notice that the steps seem slightly more simple. That's true. Sometimes in the IB, some questions will actually expect less of a detailed answer, but then you can be surprised if you look at another IB question, then the mark scheme expects a more detailed answer. So I think for safety, if you can remember these steps here, any IB question, you should get full marks, okay? So I hope this video was useful. It is a hard topic, I agree, but take it step by step, slowly, and hopefully you can get it.